cell organelles. Endoplasmic reticulum. Within the cytoplasm of most animal cells is an extensive network reticulum of membrane limited channels, collectively called endoplasmic reticulum (ER). Some portion of endoplasmic reticulum membrane remain continuous with the plasma membrane and the nuclear envelope. The outer surface of rough endoplasmic reticulum has attached ribosomes, where a smooth endoplasmic reticulum do not have attached ribosomes. Functions of smooth endoplasmic reticulum include lipid metabolism, bath catabolism, and anabolism. They synthesize a variety of phospholipids, cholesterol, and steroids. Glycogenolysis, degradation of glycogen being polymerized in the cytosol, and drug detoxification by the help of cytochrome P450. Darnell et al. 1986. On their membranes, rough endoplasmic reticulum contains certain ribosome-specific transmembrane glycoprotein called ribopurines 1 and 2, to which are attached the ribosome while engaged in polypeptide synthesis. As a growing secretory, polypeptide emerges from ribosome. It passes through the rough endoplasmic reticulum membrane and gets accumulated in lumen of rough endoplasmic reticulum. Here, these polypeptide chains undergo tailoring, maturation, and molecular folding to form functional secondary and tertiary protein molecules. The rough endoplasmic reticulum pinches up certain tiny protein-filled vesicles which ultimately get fused to cis Golgi. Rough endoplasmic reticulum also synthesizes membrane proteins and glycoproteins which are translationally inserted into the rough endoplasmic reticulum membrane. Thus, endoplasmic reticulum is the site of biogenesis of cellular membranes. Golgi apparatus or Golgi bodies. It is cup-shaped organelle which is located near the nucleus in many types of cells. Golgi apparatus consists of a set of smooth cisternae, for example, closed fluid-filled latent membranous sacs or vesicles, which often are stacked together in parallel rows. It is surrounded by spherical membrane-bound vesicles, which appear to transport proteins to and from it. Golgi apparatus consists of at least three distinct classes of cisternae: cis Golgi, median Golgi, and trans Golgi, each of which has distinct enzymatic activities. Synthesized proteins appear to move in the following direction: rough endoplasmic reticulum, cis Golgi, median Golgi, trans Golgi, secretory vesicles. Cortical granules of egg lysosomes or peroxisomes. Thus, the size and number of Golgi apparatus and acyl indicate the active metabolic, mainly synthetic state of that cell. Plant cells contain many freely distributed subunits of Golgi apparatus called Ribosomes, secreting cellulose and pectin for cell wall formation during the cell division. Generally, Golgi apparatus performs the following important functions: the packaging of secretory material, enzymes, mucin, lactoprotein of milk, melanin pigment, etc., that are to be discharged from the cell. The processing of proteins, glycosylation, phosphorylation. Sulfation and selective proteolysis. The synthesis of certain polysaccharides and glycolipids. The sorting of proteins destined for various locations, lysosomes, peroxisomes, etc. In the cell, the proliferation of membranous elements for plasma membrane, formation of the acrosome of the spermatozoa. Lysosomes. The cytoplasm of animal cell contains many tiny spheroid or irregular shaped membrane bounded vesicles known as lysosomes. The lysosomes are originated from Golgi apparatus and contain numerous about 50 hydrolytic enzymes. For example, acid phosphatase that is cytochemically identified for intracellular and extracellular digestion. They digest the material taken in by endocytosis such as phagocytosis, endocytosis, and phagocytosis. Part of the cell by autophagy and extracellular substances, lysosomes have a high acidic medium, and this acidification depends on ATP dependent portion pumps which are present in the membrane of lysosomes, which accumulate protons H plus inside the lysosomes. Lysosomes exhibit great polymorphism. There are following four types of lysosomes: primary lysosomes, storage granules; secondary lysosomes, secondary lysosome, digestive vacuoles; residual bodies, and autophagic vacuoles. The lysosomes of plant cells are membrane-bounded storage granules containing hydrolytic digestive enzymes. Large vacuoles of parenchymatous cells of corn seedlings proteins are aleurone bodies and starch granules of cereal and other seeds. Cytoplasmic vacuoles. The cytoplasm of many plant and some animal cells, ciliate protozoans contain numerous small or large-sized hollow liquid-filled structures, vacuoles. These vacuoles are supposed to be greatly expanded endoplasmic reticulum or Golgi apparatus. The vacuoles of animal cells are bounded by a lipoproteinous membrane and their function as the storage, transmission of the materials, and the maintenance of internal pressure of the cell. Vacuoles of the plant cells are bounded by a single semi-permeable membrane known as tonoplast. These vacuoles contain water, phenol, flavonols, anthocyanins, blue and red pigment, alkaloids, and storage products such as sugar and proteins. Peroxisomes. These are tiny cellular membrane bound organelles containing a crystal core enzymes such as urate oxidase, peroxidase, deamino oxidase, and catalase. For example, liver cells and kidney cells. These enzymes are required by peroxisomes and detoxification activity in the metabolism or production and decomposition of hydrogen peroxide or H2O2 molecules, which are produced during neutralization of certain superoxides. The end products of mitochondrial or cytosolic reactions, peroxisomes, are also related with beta oxidation of fatty acids and thermogenesis like the mitochondria and also end degradation of amino acids and green leaves of plants. Peroxisomes carry out the process of photorespiration. Lysosomes. These organelles develop and germinating plant seed, castor bean, or racemes to utilize stored fat of the seed to metabolize the triglycerides. Glycosomes consist of an amorphous protein matrix surrounded by a limiting membrane. The membrane of glycosomes originates from the endoplasmic reticulum and their enzymes are synthesized in the free ribosomes and the cytosol. Enzymes of glycosomes are used to transform the fat stores of the seed into carbohydrates by way of glycosylate cycle. Mitochondria, powerhouse of the cell. Mitochondria are oxygen-consuming, ribbon-shaped cellular organelles of immense importance. Each mitochondrion is bounded by two unit membranes. The outer mitochondrial membrane resembles more with the plasma membrane in structure and chemical composition. It contains proteins that render the membrane permeable to molecules having molecular weight as high as 10,000. Inner mitochondrial membrane is rich in many enzymes, coenzymes, and other components of electron transport chain. It also contains proton pumps and many permease proteins for the transport of various molecules such as citrates, ADP phosphate, and ATP. Inner mitochondrial membrane gives out finger-like outgrowths cristae or cisternae toward the lumen of mitochondria and contain tenous ricket-shaped F particles which contain ATPase enzymes for ATP synthesis. Mitochondrial matrix, which is the liquid colloidal area encircled by the inner membrane, contains the soluble enzymes of Krebs cycle, which completely oxidize the acetyl coenzyme A, an end product of cytosolic glycolysis and mitochondrial oxidative decarboxylation to produce carbon dioxide, water, and hydrogen ions. Hydrogen ions produce the molecules of NAD and FAD, for both of which pass on hydrogen ions to respiratory or electron transport chain, where oxidative phosphorylation takes place to generate energy-rich ATP molecules. Since mitochondria act as the powerhouses of the cells, they are abundantly found on those sites where energy is earnestly required, such as sperm cell, muscle cell, liver cell, up to 1600 mitochondria, macrovilli, oocyte, more than 3 lakh mitochondria, etc. Mitochondria also contain in their matrix single or double, circular, and double-stranded DNA molecules called mtDNA, and also the 55S ribosomes called mitoribosomes. Since mitochondria can synthesize 10% of their protein and their own protein synthetic machinery, they are considered as semi-autonomous organelles. Mitochondria may also produce heat, brown fat, and accumulate iron-containing pigments, heme, ferritin, ions of Ca2 plus and HPO4, are phosphate, for example, osteoblasts of bones are of proteins. Plastids. Plastids occur only in the plant cell. They contain pigments and many synthesize and accumulate various substances. Plastids are of the following types. Leucoplasts are colorless. Plastids of embryonic and germ cells lacking thylakoids and ribosomes. Amyloplasts produce starch. Proteinoplasts accumulate protein. Oleosomes are leoplasts, store fats and essential oils. Chromoplasts contain pigments molecules and are colored organelles. Chloroplasts impart a variety of colors to plant cells, such as red color in tomatoes, red chilies, and carrots, various colors to petals to flowers, and green color to many plant cells. The green colored chromoplasts are called chloroplasts. They have chlorophyll pigment and are involved in the photosynthesis of food and so act like the kitchen of the cell. Chloroplasts have diverse shapes in green algae, but are round oval or discoid in shape in higher plants like mitochondria. Each chloroplast is bounded by two membranous envelopes, both which have no chlorophyll pigment. However, unlike mitochondria, there occur third system of membranes within the boundary of inner membrane called grana. The grana form the main functional unit of chloroplast and are both in the homogeneous matrix called the stroma. Stroma contains a variety of photosynthetic enzymes and starch grains. Grana are stacks of membrane-bounded flattened discrete sacs arranged like neat piles of coins. A chloroplast contains many such interconnected grana on which are located various photosynthetic enzymes and molecules of green pigment, chlorophyll, and other photosynthetic pigments to trap the light energy. They contain DNA ribosomes and complete protein synthetic machinery. Ribosomes. Ribosomes are tiny, spheroidal, dense particles of 150 to 200 angstrom diameter that contain approximately equal amount of RNA and protein. They are primarily found in all cells and serve as a scaffold for the ordered interaction of the numerous molecules involved in protein synthesis. Ribosome granules may exist either in the free state in the cytosol, for example, basal epidermal cells, are attached to rough endoplasmic reticulum, pancreatic acinar cells, plasma cells, or antibody secreting lymphocytes, osteoblasts. Ribosomes have a sedimentation coefficient of about 80s and are composed of two subunits, namely 40s and 60s. The smaller 40s ribosomal subunit is prolate ellipsoid in shape and consists of one molecule of 80s ribosomal RNA or rRNA and 30 protein named as S1, S2, S3 and so on. The larger 60S ribosomal subunit is round in shape and contains a channel through which growing polypeptide chain makes its exit. It consists of three types of ribosomal RNA molecules: 28S ribosomal RNA, 5.8 ribosomal RNA, and 5S RNA, and 40 protein named L1, L2, L3, and so on. Microtubules and microtubular organelles. With rare exceptions such as human erythrocyte, microtubules are found in the cytoplasm of all types of eukaryotic cells. They are long fibers of indefinite length, about 24 nanometer in diameter, and cross secretion. Each microtubule appears to have a dense wall of 6 nanometer thickness and a light or hollow center in cross section. The wall of microtubule is made up of 13 globular subunits called protofilaments, about 4 to 5 nanometer in diameter. Chemically, microtubules are composed of two kinds of proteins subunits: alpha tubulin, tubulin A, and beta tubulin, tubulin B, each of MW 55,000 Daltons. The wall of microtubule is made of a helical array of repeating alpha and beta tubulin subunits. Assembly studies have indicated that structural unit is an alpha beta dimer of 8 nanometer length. Thus, in each microtubule, there are 13 protofilaments, each composed of alpha V dimers that run parallel to the long axis of the tubule. The repeating unit is an alpha beta heterodimer, which is arranged head to tail within the microtubule, that is alpha beta alpha beta alpha beta. Thus, all microtubules have defined polarity. Their two ends are not structurally equivalent. Microtubules undergo reversible assembly, disassembly, polymerization, depolymerization depending on the need of the cell or organelles. Their polymerization is regularized by certain MAPs or microtubule associated proteins. The assembly of microtubules involves preferential addition of subunits alpha beta dimers to one end of tubule called A end or net assembly end. The other end of the tubule is called D end or net assembly end. Such an assembly involves the hydrolysis of GTP to GDP. Thus, assembly of tubulin in the formation of microtubule as a specifically oriented and programmed process rheos, basal bodies, and centromeres of chromosomes are the sites of orientation for this assembly. Calcium and calmodulin, an acidic protein having four Ca2 plus binding sites, are some other regulating factors in the in vivo polymerization of tubulin. Certain drugs such as colchicine and vinblastine are found to block the polymerization of tubulin. The following cell organelles are derived from special assemblies of microtubules: cilia and flagella. Ciliary and flagellar cell motility is adapted to liquid media and is executed by minute specially differentiated appendices called cilia and flagella. Both of these organelles have very similar structure. They differ mainly in size and number. Flagella are longer and fewer in number, while cilia are short and numerous. Cilia are used for locomotion in isolated cells such as certain protozoans, paramecium, or to move particles in the medium, as in air passages and oviducts. Flagella are generally used for locomotion of cells such as spermatozoon and euglena protozoon. All cilia and flagella are built on a common fundamental plan, a bundle of microtubules called the axoneme, 1 to 2 nanometer in length and zero point 0.2 micrometer in diameter, are surrounded by a membrane that is part of plasma membrane. The axoneme is connected with the basal body, which is an intracellular granule lying in the cell cortex and which originates from the centrioles. Each axoneme is filled with ciliary matrix, in which are embedded two central cingulate microtubules, each with the 13 protofilaments and 9 outer pairs of microtubules called doublets. This recurring motif is known as the 9 plus 2 array. Each doublet contains one complete microtubule called a subfiber containing all the 13 protofilaments. Attached to each a subfiber is a B subfiber with 10 protofilaments. Subfiber A has two dynein arms which are oriented in a clockwise direction. Doublets are linked together by Nexian links. Each subfiber A is also connected to the central microtubule. Doublets are linked together by Nexian links. Each subfiber A is also connected to the central microtubule by radial spokes terminating in fork-like structures called spoke knobs or heads. Propulsion by both cilia and flagella is caused by bending at their base. Cilia move by a whip-like power stroke fueled by hydrolysis of ATP, followed by recovery stroke. Flagellar movement is also powered by ATP hydrolysis and contracts to cilia. They generally move by waves that emanate from the base and spread outward toward the tip. Basal bodies and centrioles. Basal bodies and centrioles are similar in structure and function, but act as nucleating centers from which microtubules grow. Centrioles are cylinders that measure zero point 0.2 micrometer to zero point 0.5 micrometer. The cylinder is often on both ends unless it carries calcium or flagellum. Then it is called basal body or kinetosome. The wall of a centriole has 9 groups of microtubules arranged in a circle. Each group called blade is a triplet formed of 3 tubules A, B, and C that are skewed toward the center. Tubule A has 13 protofilaments, while tubule B and C have only 10 protofilaments. Tubule A has 13 protofilaments while tubule B and C have only 10 protofilaments each. There are no central microtubules in the centriole and no dynein arms like the cilia. However, triplets are linked by connectives. The procentriole or daughter centriole is formed at right angle to the centriole and is located near the proximal end of the centriole. Both centrioles are found in a specially differentiated region, the centrosome, cell center, or centrosphere. The centrosome is juxtanuclear, juxta, near, and firmly attached to the nuclear envelope. At the time of cell division, two pairs of centrioles are formed and form the spindle of microtubules, which help in separation and movement of chromosomes during concluding stages of cell division. Nucleus, most important organelle of the cell. The nucleus is centrally located and spherical cellular component which control all the vital activities of cytoplasm and carries the hereditary material, the DNA, and it. The nucleus consists of the following three structures. Chromatin. Nucleus being the heart of every type of eukaryotic cell contain the genes, the hereditary units. Genes are located on chromosomes which exist as chromatin network and the non-dividing cell during interphase. The chromatin has two forms. Euchromatin as the well-dispersed form of chromatin which takes lateral DNA strand and is genetically active. It is involved in gene duplication, gene transcription, DNA dependent RNA synthesis, and phenogenesis or phenotypic expression of a gene through some type of protein synthesis. Heterochromatin is the highly condensed form of chromatin which takes dark DNA stain and is genetically inert. Such type of chromatin exists both in the region of centromere called constitutive heterochromatin and in the sex chromatin called facultative heterochromatin and is replicating one. Chemically, the chromatin contains a single DNA molecule equal amount of 5 basis types of stone protein, some RNA molecules, and variable amount of different types of acidic proteins. In fact, the chromatin has its unit structure in the form of nucleosomes. The chromatin binds strongly to the inner part of nuclear lamina, a 50 to 80 nanometer thick fibrous lamina lining the inner side of the nuclear envelope. Nuclear lamina is made of three types of protein, namely lamine A, B, and C. Lamine proteins are homologous in structure to IA proteins and serve the following functions. They anchor part of interface chromatin to the nuclear membrane. They tend to interfere with chromatin condensation during interphase of cell cycle. Lamines may play a crucial role in assembly of interphase nuclei after each mitosis. Nuclear envelope and nucleoplasm. Nuclear envelope comprises two nuclear membranes, an inner nuclear membrane which is lined by nuclear lamina and an outer nuclear membrane which is continuous with rough endoplasmic reticulum. At certain points, the nuclear envelope is interrupted by structures called pores or nucleophores. Nucleophores contain octagonal pore complexes which regulate exchange between the nucleus and cytoplasm. The number of nucleophores is found to be correlated with transcriptional activity of the cell. For example, in the frog Xenopus lewis oocytes, which are very active in transcription, have 60 pores micrometer square and up to 30 million pore complexes per nucleus, whereas frog mature erythrocytes inactive in transcription have only about 3 pores micrometer square and a total of only 150 to 300 pores per nucleus. The nuclear envelope binds the nucleoplasm, which is rich in those molecules which are needed for DNA replication, transcription, regulation of gene actions, and processing of various types of newly transcribed RNA molecules, transfer RNA, messenger RNA, and other types of RNAs. Nucleolus. Nucleus contains in its nucleoplasm conspicuous, darkly stained circular suborganelle called nucleus. Nucleus lacks any limiting membrane and is formed during interphase by the ribosomal DNA or DNA of nuclear organizer. Nucleolus is the site where ribosomes are manufactured. It is here where ribosomal DNA transcribes most of RNA molecules, and these molecules undergo processing before their stepwise addition to 70 types of ribosomal protein to form ribosomal subunits. Difference between plants and animal cells. Animal cells are generally small in size. Plant cells are larger than animal cells. Cell wall is absent in animal cell. The plasma membrane of plant cell is surrounded by a rigid cell wall of cellulose. Except the protozoan euglena, no animal cell possess plastids. Plastids are present in plant cells. In animal cells, vacuoles in animal cells are many and small. In plant cells, most mature plant cells have a large central shaped vacuoles. Animal cells have a single highly complex and prominent Golgi apparatus. Plant cells have many simpler units of Golgi apparatus called dictysomes. Animal cells have centrosome and centrioles. Plant cells lack centrosome and centrioles.